everyone, in this little video I'm going to be showing you a model of the Arabinose operon and how it is regulated. So this region down here is actually the Arabinose operon. Here we have the PBAD promoter to which the RNA polymerase will bind, and then over here we have the three genes that are part of the Arabinose operon, and each of these genes codes for an enzyme to break down arabinose. Upstream here we have the different regulatory sequences. So you'll remember, hopefully, that the Arabinose operon is regulated by a protein called ARAC. So the ARAC protein, in addition to regulating the Arabinose operon, also regulates its own transcription. And so that's what I've shown here with these little binder clips. I'm using binder clips as the ARAC. I've shown that the ARAC is bound here at the ARA01 operator, and when there's a homodimer bound here, it prevents additional production of the ARAC protein. So it regulates itself in addition to regulating the Arabinose operon. But when it regulates the Arabinose operon, we're going to have one copy of the ARAC protein that's going to bind here to the ARAI site. Then we're going to have a second copy of the ARAC protein that's going to bind here to the ARA02 site. Then these two are going to connect together, which happens when the DNA is folded around. Hang on, I'm having an issue. Okay, so the DNA folds around, and then, oopsie, these two ARAC proteins are going to bind to each other to be a homodimer. And so they are keeping the DNA folded like this. Consequently, the RNA polymerase is unable to locate and bind to the PBAD promoter, which is kind of mushed up here in this folded up DNA. So when there is no arabinose, then the two ARACs are going to be bound to ARAI and ARA02, and they're going to form the DNA into this fold, preventing transcription of the Arabinose operon. When arabinose is present, this is our little Arabinose guys, then arabinose is going to bind the ARAC, and then what actually happens is the ARAC changes shape, which we already know. Anytime an effector binds, the ARAC changes shape. So we're going to have some arabinose bind down there. And since these are binder clips, I can't really change their shape, but we're going to have an arabinose bind here. We'll put it on the end. And then we're going to have another ARAC bind here. So the consequence of the ARAC changing shape is that now they are more flexible, and so two of the ARAC molecules actually will fit here in the ARAI region, and they will bind to each other. Consequently, the ARACs are happy because they are interacting in the pair, but because of the way that their shape is changed, when the arabinose binds, they can both bind here at the ARAI, allowing the DNA to lie flat. However, the RNA polymerase still is not prepared to bind to the PBAD promoter. What is it missing? It is missing this other activator here. So you'll remember that in the cytoplasm we have this inactive CRP or CAP activator. And this activator is waiting for the cyclic AMP to come and bind to it. The cyclic AMP tells the cell that there is not very much glucose available. So when the cyclic AMP comes and binds, then the shape of the CRP activator is changed. Then the activator is prepared to bind here to the activator binding site. Once both this activator is bound here to the cap site, and there are two of these ARAC molecules with arabinose bound here, making the DNA flat, then the sigma factor and RNA polymerase can bind here to the PBAD promoter and then move down the operon and transcribe the three genes. So that's how the Arabinose promoter, the Arabinose operator, the Arabinose operon is regulated.